Well, good afternoon. I'd like to thank the organisers for inviting me to speak, albeit from my garage in my home in Northern California. My name is Patrick Phillips. I'm an assistant professor at the UCSF Centre for TB, and most of my research involves the design, conduct and analysis of randomised trials to improve the treatment of TB. My title today is Advancing TB Regimens from Phase II to Phase III, Navigating the Land of Expectations. I'm going to be talking about trial design in this space here, this Phase II area. On the left, we have the Phase I domain, early indication of efficacy of individual drugs and limited data on combinations. On the right, we have the Phase III trial, gold standard for confirmation of efficacy. And I'm going to be talking about this middle phase here, which is often called middle development, how we move through Phase II and identify evidence to support a future Phase III trial. For the purpose of this talk, I'm going to christen this the Land of Expectations. And I've taken this from the famous children's book that my son was reading a few months ago, The Phantom Tollbooth by Norman Juster. And here the protagonist is transported through this phantom tollbooth and arrives in a different world. And he sees this sign, "Welcome to Expectations," says a carefully lettered sign in a small house at the side of the road. Information, predictions and advice cheerfully offered. Where he meets the weatherman, who helps him along his travels. And so today I'm talking about trial design and about how we design trials that aim to successfully navigate this land of expectations to identify the best regimens to take forward to Phase III. And my context here is drug development for tuberculosis, and I want to say two things about TB. The first of which is that TB is a global health emergency. This shows the trends in incidence over the last 20 years, not a lot of progress. If we think about the deaths so far in 2020, there's been approximately 280,000 deaths due to tuberculosis, half as many due to HIV. And you can see at the bottom there how this compares to the 3,282 who have died of COVID-19 this year. The second thing I want to say is that in 2020, there are new drugs ready for evaluation in the land of expectations. You can see on the left the 2012 new TB drug pipeline, quite sparse, and a very different picture here in 2020, a number of agents ready for clinical trial evaluation. TB is not HIV. Here is an example showing the clinical development of Dolutegravir, and I want to highlight the primary outcomes here. You can see even from Phase IIA, where the primary outcome was reduction in plasma HIV-1 RNA over 10 days, right through Phase IIB and Phase III, virologic suppression at week 16 in Phase IIB and week 48 in the Phase III trial. So a clear and consistent primary outcome throughout development. TB is different. The bacterial course of TB disease is not well understood. This figure illustrates how we think Phase II and Phase III outcomes are related. On the left here, we have the Phase II outcomes. And on the right, these are the Phase III outcomes. And the key point to note here is that what's happening here, how the Phase II and the Phase III outcomes relate, is below the limit of detection. So we have some theories about what's happening down there, but it's true that surrogacy of these Phase II outcomes is not established. This is work that I did a few years ago, looking at whether the treatment effect on the 2-month culture or the treatment effect on the 3-month culture is able to predict the treatment effect on a long-term durable outcome. And the key thing to note is there's a lot of spread around the lines. And whilst the 3-month outcome may be better, there's a lot of spread, meaning that the conclusions you would draw from month 3 culture results may not quite be the same as what you would get from the long-term outcome in terms of differences between treatments. Here is the outline of my talk today as we navigate the land of expectations. The first question is, what is the right dose? The second question, how do we make progress when surrogate endpoints are not established? And I'm going to talk about the Phase IIC design. Thirdly, which are the best combination regimens to advance? How do we go about answering that question? Fourth, what is the right duration? Fifth, should all patients receive the same treatment? And finally, bringing it all together, platform trials and global public-private networks. So first of all, what is the right dose? And here, PK/PD modeling is essential at every intersection. Here are two examples, rifapentine at the top and rifampicin at the bottom, showing how state-of-the-art PK/PD modeling has shown the benefits of increased doses of the rifamycins. So what are the trial designs that we might consider for single drug dose finding? Well, the first is a concurrent randomized comparison of multiple doses of a drug added to a fixed backbone combination. This has been done in TVTC study 29X with rifapentine and was planned for the PanACEA Sudoku study looking at dose finding for sertazolid. An alternative is looking at sequential dose escalation cohorts, as was done in the PanACEA HR1 study, looking at rifampicin dosing in 7 cohorts, sequential cohorts from 10 all the way up to 50 milligrams per kilogram. A third alternative is really thinking more innovatively about this and thinking about dose escalation in the context of an adaptive dose response modeling design. Here a nice example is the PanACEA BTZ-043 Phase IIA study. This is using a Bayesian continual reassessment method to model the dose response relationship and use a quantitative model to recommend the best dose to take forward in the next cohort. When we have multiple drugs, we need to think about a different approach, and when we can consider a surface randomization, where we're jointly optimizing doses of drugs across several drugs. Here the idea is that you randomize not just to a dose of a single drug, but across the matrix of doses across, in this example, 2 drugs. This is an efficient design and allows you to estimate the surface with very few patients per combination. I don't know that this design has been used in TB yet, but I think it's a useful design that we could use to optimize dose finding across multiple drugs. Second question, how do we make progress when surrogate endpoints are not established? And I want to talk about the Phase IIC design here. This is a representation of the classic Phase IIB design for TB regimen development. Standard therapy for TB is 6 months of therapy, which is made up of a 2-month intensive phase followed by a 4-month continuation phase. And so the standard approach is to, in this example, 3 experimental arms. Standard approach is to replace the intensive phase with 2 months of our experimental treatments, experimental regimen, but continue the standard continuation phase after that 2 months to make up a total of 6 months of therapy for all patients on all arms. The Phase IIC design takes a different approach. Here, we're studying the intended duration of the experimental arms. So the experimental regimen is given for 3 or 4 months, whatever is going to be targeted, treatment stops, and patients are followed up for long-term outcomes, up to 12 months post-randomization. And here the principle is that we're studying the intended duration of regimens in Phase 2 rather than waiting for that question in Phase 3. There is going to be potentially a slight increase in sample size and cost due to longer follow-up than might have been done in a Phase 2B trial. But the key here is this is richer data collected in this middle development phase to inform better decision-making for Phase 3. So the primary endpoint would be bacterial decline during treatment. We would have adequate power to detect differences between regimens with a sample size of 80 to 100 per arm, typical of a Phase 2B trial. But as I've already said, this is only an intermediate endpoint that only really partially reflects long-term clinical outcome. Our secondary outcome in these studies is long-term relapse-free cure. Now, the trial is underpowered to detect differences in relapse rates, but this is the more patient-relevant clinical outcome. The fundamental research question here is, will this shorter regimen have efficacy at least as good as the 6-month control in a future non-inferiority Phase 3 trial? Remember, this is about navigating this land of expectations with a view to moving on to a Phase 3. And this sort of question naturally fits into the Bayesian framework. And so what we have here is a Bayesian calibration curve, and this allows us to make predictions about the future Phase 3. So the curves here are plotted for a flat prior, the dashed lines are a skeptical prior, the solid lines. If we assume that we're targeting a 8% relapse rate for a future Phase 3, and we've got 100 patients per arm, we're looking at a green solid line in the middle there. And so we might read off the results from our Phase IIC trial. If we have an observed 3% relapse rate, this means that there's a 92% chance of success in Phase 3. If we have an observed 6% relapse rate, well, this means there's only a 62% chance of success in Phase 3. And we proposed a traffic-like system for this predictive probability of Phase 3 success. If the predicted probability exceeds 0.95, that's a green light, that's good evidence to move forward to Phase 3. If it's less than 0.5, that's a red light. That's unlikely to be a viable regimen in Phase 3. And you can see there's between 0.95 and 0.5, there's some questions about looking at other outcomes and about safety. We did some work looking to see whether a Phase IIC trial prior to the Rifaquin Phase III trial would have added value. Rifaquin's a nice example because there was a 6-month intermittent regimen that was shown to be non-inferior and a 4-month regimen that was not non-inferior. And so what we can see here across 10,000 simulated Phase IIC trials is the inferior regimen would have got the red light, the Phase IIC trial results would have recommended not going forward. On the other hand, with 100 patients per arm, the successful regimen in 43% of simulations would have got a green light, and in 96% of simulations, the predicted probability would have been above 0.75, showing that this would have been a useful tool prior to the Rifaquin Phase III trial. This Phase IIC design has not been implemented yet in a trial, but is being considered for a number of trials that are being planned. The PanACEA STEP Phase IIC trial, the ACTG CLOFAST trial, the TBTC CRUSH TB trial, trials that are hoping to start soon. There are extensions that we can think of. We can think about joint Bayesian model of intermediate and long-term outcomes, and we can take a step further in thinking about putting prior distributions on other Phase III parameters. For example, the control arm efficacy or parameters related to the patient population. This would allow us to really improve what that predicted probability means and think about different types of Phase III trials and the likelihood of success in different types of Phase III trials. If we have a design that allows us to make progress when surrogate endpoints are not established, how do we decide which arms to advance? And I want to now talk about treatment selection designs. History has shown us that 3 or 4 drug combination regimens are necessary in TB to affect, cure, and prevent drug resistance. So if we take the 4 or 5 current drugs we have available, or repurposed drugs, add to that the 8 to 10 new drugs that are coming through, it's easy to see that could lead to many possible combination regimens that we could evaluate. And so treatment selection designs are going to be essential to sensibly identify the most promising combinations. I'm going to talk about 2, the multi-arm, multi-stage MAMS design and Bayesian response adaptive randomization. So first, looking at the MAMS design. This is a multi-arm Phase 2 or seamless Phase 2/3 trial with several planned interim analyses. It involves individual pairwise comparisons against the control arm, and then arms without sufficient evidence of benefit compared to the control are dropped using a pre-specified critical value or hurdle, and the hurdles are raised at each interim analysis. This is an example of a 6-arm trial, 5 experimental arms. On the vertical axis, this is the treatment effect versus control, and this is just an example of what might happen. You can see at the end of the first stage, the first interim analysis, all the arms are above the hurdle, and so continue to the second stage. 2 arms do not continue into the third stage, and then we have 2 of the 5 arms that continue into the 4th stage. You can see the hurdles increase at each point, and only the 2 arms reach the end of the study ready for the final analysis. We implemented this design in the PanACEA MAMS-TB Phase IIB trial, bringing together 3 drug development programs into one MAMS trial. We included a novel compound SQ109, an existing drug, higher doses of rifampicin, and a repurposed drug, moxifloxacin. We had 1 interim analysis where 2 of the 4 experimental arms recruitment has stopped, 2 arms continued, and in the final analysis we're able to show that this 1 arm with a 3.5-fold increase in the dose of rifampicin did result in shorter time to culture conversion as compared to control. So it's successfully implemented in this trial. An alternative design is the Bayesian response adaptive randomization. This also is a multi-arm Phase 2 trial or seamless Phase 2/3 trial. And here, instead of interim analyses, the allocation ratio, the randomization allocation ratio, is adapted as data accrue to randomize more patients to the best performing arms. This means that as the trial progresses, fewer patients are allocated to the least effective arms. The adaptive algorithm uses intermediate outcomes to predict final outcomes, but also the model, a predictive model here, learns as final outcome data become available, and so is updated as data accrues. A nice example of this is the NTB clinical trial for rifampicin-resistant TB, 5 experimental arms. And this just shows you an illustration of how the design might work. So initially, the trial begins with equal allocation across the 6 arms. As 8 and 39-week responses become available, a model is used to predict the 73-week response and adapt randomization. In this example, more patients to arm 3 and the control. And as more data accrue, and particularly as the 73-week data becomes available, the model is updated, and again the allocation ratio is updated throughout the trial, so that more and more patients get put on the better arms, in this case arm 3, and fewer patients get allocated to the arm that's not doing as well, arm 2. And the trial finishes when the sample size is reached, at which point the most patients have been put in the best performing arms and the control, which increases the power for this for information about the best arms, with fewest patients on the arms that are not doing as well. So it's efficient in that regard. So in summary, these designs consist of pre-specified design and critical values, so it's a single trial protocol. And I think of it in terms of resource allocation. So we're focusing resources on regimens of most interest and moving resources away from those that are of least interest. And it answers a more relevant public health question, not is this drug or regimen effective, but instead, among many, which the most effective interventions. It doesn't require a perfect surrogate for resource allocation because all patients are being followed for the long-term clinical endpoint in both these designs, but it does help. And certainly better and definitely more rapid biomarkers are needed to improve how efficient these designs are. If there are biomarkers that are available sooner, then the allocation ratio can be adapted sooner, or poorly performing arms can be dropped sooner in the trial to increase efficiency. So the next question is, what is the right duration? And this is not a question that needs to be considered in HIV drug development, where treatment is for life, but is a critical question in TB. Failing to identify the right duration in phase 2 leads to a dilemma. So either you take a big risk in phase 3, take one duration into phase 3, an example being the 4-month fluoroquinolone regimens that were studied in Remox, Rifago, and Oflatab, and were shown to be non-inferior. Now, the moxifloxacin regimen was probably a 5-month regimen based on retrospective analysis, admittedly, but it was not possible to learn this from the Remox trial because only the 4-month regimen was studied. The alternative is to do a big phase 3 trial, select many durations in phase 3. An example of this would be the STAMPEDE trial that had 3 durations of the same regimen. And so the question is, can we do better in phase 2? So let's go back to look at what we know about current first-line therapy. And we actually have reasonable data on the odds of durable cure with different durations of first-line therapy. We can see here there's monotonic increase. We can see it's approximately linear over the middle region, and it flattens out with long durations, meaning that the longer a therapy is given, it's not going to increase the odds of cure dramatically. So the question is, can we exploit this knowledge for the development of new regimens? And so this is really what the duration-randomization design is about. The approach is to randomize to multiple durations of a novel regimen or to the control, and work has been done that shows 5 or more durations is optimal. And then the approach would be to fit a duration response model to these data in the same way as you might fit a dose response data to data looking at different doses of the same drug. A fractional polynomial of degree 2 is sufficiently flexible for the set of plausible shapes, and you can see at the top there on the right 2 sort of different shapes that might be plausible, and the fractional polynomial describes both of these curves reasonably well. The red lines corresponding to simulations from the simulation study. My example on the left is showing what this might look like in TB. This is only 3 durations as an example, and I've just fitted a straight line for simplicity. The vertical axis is the odds ratio of success as compared to control, and what you can see is that when the region, the confidence region, is completely above our pre-specified margin of non-inferiority, the dotted line, the horizontal dashed line, excuse me, you can identify the shortest non-inferior duration, which in this example is 27.7 weeks. And there's the potential also to identify the shortest superior duration. In this example, is 38.3 weeks when the fitted region is completely above an odds ratio of 1. We're making progress through the land of expectations, but we need to ask ourselves, should all patients receive the same treatment? Here is a summary of relapse rates across a range of durations of between 2 and 4 month durations, so shorter than standard of care. Most of these relapse rates are unacceptable. You can see 15%, 16%, 18, 20, 22%, and none of these regimens are used or recommended. But if we look at the other side of the coin and compare this to untreated tuberculosis, we expect there's only about a 30% self-cure, we can see that all of these regimens cure the vast majority of patients with TB. Most cases well above 80%, some cases 85, close to 90%, beyond 90%. And the challenge in the past has always been to identify those patients that can be cured with these short regimens. And recent analyses have provided evidence to show that we probably can prospectively stratify patients by risk of relapse. So patients with low bacillary loads and absence of cavities are at low risk of relapse. And in this subpopulation, the short regimen was shown to be non-inferior to the standard of care. On the other hand, those with high bacillary load, cavitary disease, low BMI are at higher risk of relapse and probably need longer treatment to be cured. So this tells us that trial level efficacy is driven by the subgroup of participants with a high risk of relapse. So what are the implications for trial design? Well, one option is to enrich for participants at high risk of relapse. This means a higher number of relapses observed in a Phase IIC, which gives greater power for discriminating between regimens. An alternative is to assign different durations for different risk strata. So patients in a low risk strata, we can pursue radical treatment shortening with no reduction in efficacy. At the same time, patients in the high risk strata, we can target longer treatment regimens for increased efficacy. So finally, bringing it all together, I want to talk about platform trials and global public-private networks. So what is a platform trial? A platform trial involves multiple interventions compared against a common control. It's a single master protocol, and there's often an adaptive design that provides a platform for arms to enter and exit at different points during the trial. And this results in efficiencies in recruitment, staffing, regulatory IRB approvals, and other efficiencies in having a single master protocol. A nice example of this is the STAMPEDE trial, which is a MAMS trial in prostate cancer. Now, when the trial was started, there were 6 arms, 5 experimental arms with 1 standard of care control. After interim analysis, you can see in 2011, recruitment has stopped to 2 arms and continued to the other 3 intervention arms. But along the way, new arms were added to the trial through protocol amendments to allow multiple different combination regimens in this setting to be evaluated in the same trial, in the same set of sites where contracts for staffing were set up in the same trial protocol through protocol amendments, through regulatory IRBs. And what this meant was that you can see 3, 6, 9, 11 interventions were evaluated over the course of 15 years in a single trial in what would have taken decades longer to do if it had been 11 separate trials. A nice example of the efficiencies that you see in the context of a platform trial. So what might this look like in TB? Here's an example of what a platform trial could look like. Here, eligible patients with drug-sensitive TB are randomized to one of 4 regimens or a control, but also randomized to different durations of those regimens, ranging from 8 to 24 weeks. So in the Phase IIC context here, all patients are followed for 52 weeks after randomization. We might consider using a MAMS type design, and here after the first interim analysis, we might look at the duration response model, which would, in this example, tell us that the 2 shortest durations for regimen A are not likely to be safe, not likely to be shown to be non-inferior to control in the future Phase 3. Similarly, the shortest duration for regimen C is also stopped. And so these 3 durations of these 2 regimens, recruitment stops after the first interim analysis. After a second interim analysis, more data is available. There's more information to model that duration response curve, identify further shorter regimens that are unlikely to show success in the future Phase 3. And then in the final analysis, we would employ our duration response models. And these provide selections for future Phase 3. So in this example, regimen A, a 24-week regimen, has been shown that it's likely to have success in a Phase 3 trial. This may or may not be useful to take forward to a Phase 3. Perhaps regimen A is a particularly safe regimen, or there may be other benefits in taking regimen A forwards. Alternatively, regimen B here is shown to be a more promising regimen, and the model has identified an 8 and potentially a 12-week regimen to take forward to Phase 3. So again, depending on safety signals, other considerations, it might be wise to take forward 1 or 2 durations forward to Phase 3. And you can see the same for regimens C and D. And so the idea here is that what this sort of platform trial would do is not only provide information about which regimens to take forward to phase 3, but provide some information about which are the likely durations that are most likely to be promising in a future phase 3. So guidance about which durations to take forward, as well as which regimens to take forward. Now, these sort of platform trials are only going to be possible with global collaborative public-private networks. A nice example is the TBTC Study 31 ACGG A5349 trial, where 2 US federally funded networks are working together to complete the largest ever active TB treatment trial. 2,500 patients are expecting results later this year. Another example is the Bill and Melinda Gates Medical Research Institute PanTB collaboration with 4 drug companies that was just announced a week or 2 ago. And also the EU-funded IMI Unite for TB, public-private collaborative research for phase 2 development of TB regimens. And this is a really nice example of a true public-private collaboration where the EU governments are putting money in, as well as the companies themselves, coming together, hopefully to bring together a big collaboration that is going to be able to deliver the sort of platform trials that we've been talking about today. And I just want to finish with some acknowledgements, starting with colleagues from the Centre for Tuberculosis at UCSF, particularly colleagues from the PanACEA network funded by the EDCTP. You would have seen that a number of these more novel designs are being implemented in the PanACEA network. We've had the opportunity to do that there. I want to thank colleagues from the TB Trials Consortium, colleagues from my previous institution where I worked, the MRC Clinical Trials Unit, and just to mention a few particular names, Andrew, Michael, Payam, Carol, Bob, and Martin, from whom I've learned a great deal. Thank you very much.